Hello, Morgan. Hello, Susan.
No,
All right, welcome. Oh, good.
I did find a paper just right, well, this morning. Oops, if I can find out where it landed. Oh, here we go. It's called Feedback Controlled Epithelial Mechanics, Emergent Soft Elasticity, and Active Yielding.
Oh, wow. All right, so this is feedback controlled epithelial mechanics, emergent soft elasticity, and active yielding. So this is Christina Marchetti, is a familiar name in this area. All right, then, so the abstract, let's go over that. Biological tissues exhibit distinct mechanical and rheological behaviors during morphogenesis. While much is known about tissue phase transitions controlled by structural order and cell mechanics, key questions regarding how tissue scale nematic order emerges from cell scale processes and influences tissue rheology, and that process remains unclear. So these are a minimum vertex model that incorporates a coupling between active forces generated by cytoskeletal fibers, their alignment with local elastic stress in solid epithelial tissues. We show that this feedback loop induces an isotropic nematic transition, leading to an ordered solid state that exhibits soft elasticity. Further increasing activity drives collective self-yielding, leading to tissue flows that are correlated across the entire system. Let's see. And then this remarkable state that we dub plastic nematic solid is uniquely suited to facilitate active tissue remodeling during morphogenesis. It fundamentally differs from the well-studied fluid regime, where macroscopic elastic stresses vanish, and the velocity correlation length remains finite, controlled by activity. Altogether, our results reveal a rich spectrum of tissue states, jointly governed by activity and passive cell deformability, with important implications for understanding tissue mechanics and morphogenesis.
No, there are images of the cells aligned in one direction in one part of the tissue and then in another direction in another part.
Oh, so yeah, it switches direction kind of abruptly.
Yeah, it can.
Okay.
Between one part of the tissue and the other.
Yeah.
And the cells are just aligned in one direction, and then for some reason they're aligned in another. So, but of course that can change.
Yeah. See if they have any good pictures in here or images. Okay, so this is a schematic of the active feedback loop and active extensile stress generated by cellular fibers in alignment with local elastic stress. So A is cell shape elongation. So the cells elongate along some axis here. The blue hexagons are the sort of the original shape, and then the red hexagons are the elongated shape, the elongated cells.
That's what I wanted to study in my thesis, but I had trouble enough making the thing stand up and not fall down according to the software.
Yeah.
So, yeah, that's what I wanted to do.
Yeah. And without going into their mathematical model, we have alpha and beta. We have cell deforms under active stress. That's alpha, and then that's an input, and then fiber aligns with elastic stress, which is beta. And you can see that then that leads to the cellular nematic order. So it's like if you get you have stresses, the cells deform, the fibers align with this elastic stress. So there's active stress and elastic stress components, and then that determines the nematic order. And I guess there's like some phase transition that happens in certain regions of the tissue that give it like these shifts in nematic order. So you can see it's kind of changing its orientation here. They have these little like sticks or lines underneath down here. B is a phase diagram where we have effective activity, alpha, beta on the y-axis, target cell shape index, P on the x-axis, and then the phases in this area. Isotropic solid, which is in the lower part of the diagram. Isomorphic gases, I guess. Nematic gas above it. Nematic liquid above that. Plastic nematic solid, which is this region that they say is this new thing that they've discovered in the paper. And that's actually, we'll see this in a minute. And then soft nematic solid underneath that. So plastic nematic solid is sort of this region here in green. It's high effective activity, low target cell shape index. As the target cell shape index decreases, the effective activity increases. And then this figure here at the right, C, I'm going to zoom in a little bit here. So these are the different sort of patterns that you see here. So soft nematic solid looks like the thing on the upper left. It's kind of got this swirl or these whorls, almost like fingerprints. They're all kind of they're all 5 and 6 shaped objects. They're hexagons, they're pentagons, and they're kind of in this swirl. Like you can see the swirl here, where there's this, they change direction, but like it isn't abrupt necessarily. Plastic nematic solid, it looks, has kind of this. It almost looks like a force diagram where I have my screensaver for my Mac, iMac is like this, where it's like it's got this kind of these orientations, but they kind of curve and they're kind of opposed to one another. Kind of interesting. The kind of the way the dynamics of this pattern. And then that's the one they're proposing in this paper that they've discovered. Nematic liquid is more sort of these abrupt changes with respect to the sort of the orientation, and you can see the cells underneath there. Nematic gas is like more chaotic than the nematic liquid, which is based on the yellow lines. Isotropic solid are these hexagons and pentagons that are basically, they look like they're sort of frozen in place. They look pretty uniform. And then the isotropic gas for the cells are sort of squished, but it looks similar to the isotropic solid. So the isotropic state is, pretty, looks, maybe stable, more stable. The plastic nematic solid looks, in the nematic gas and the nematic liquid look more dynamic to me. The color map is the magnitude of nematic order parameter. So the nematic order parameter is Q. The closer to 1, the more purple it is. So you see a lot of purple in the isotropic solid condition, a lot of kind of dark blue to purple in the nematic gas, some dark blue to purple in the in the nematic gas and nematic liquid, and then the soft nematic solid has these blue and purple regions only, or dark blue and purple. So you can see that they correspond to that Q parameter. Okay, let me see if they have any more details on their mathematical model. Actually, this figure 2 is interesting. This is where we have this trijunction. We have cell J and cell I, and then the cell down here. And this just shows the nematic, the parameter we just mentioned, which is this nematic order, and then S, which is shape anisotropy. So you have the order parameter Q and the anisotropy S. And you can see that stress-induced order, they kind of start at 1 on the alpha beta scale and they go up. S going up slower than Q. These are curvilinear. functions, and that describes the stress-induced order. And it's just kind of where stresses are applied to the system. And because they're already kind of joined together, they kind of push the sides of the cell in different ways, and they all kind of have the same order. Then there's intrinsic order, which is this here. This again is Q, the order parameter S, which is shape anisotropy. And so, you understand that shape anisotropy is where it's not isotropic, like you have differences in the shape. And the differences in the shape will increase as the order parameter increases in the first case, in the stress-induced order. In the intrinsic order, the order parameter and the shape anisotropy are kind of separated by about 0.5 units on the y-axis, and then S increases a little bit. It starts at 0 and it increases a little bit more curvilinearly than Q, but the shape anisotropy is low while the order parameter is high. Figure 3, these tissue snapshots of different activity you have for the isotropic solid, the soft nematic solid, and the plastic nematic solid. And you have these different values for S and Q. So the shape anisotropy in orange, the order parameter in green. When it's isotropic, the shape anisotropy is greater than the order parameter. In the nematic case, where there's a phase transition on the alpha beta axis, which is right around the value of 1 for alpha beta, there's a phase transition where shape anisotropy is lower than the phase transition. And then you have in this next, so that's for the order parameters versus alpha beta, and then for alpha beta versus mean velocity and T1 transition rate, you have the isotropic solid, the soft nematic solid. Those have the same sort of near zero mean velocity for both the shape anisotropy and order parameter. And then towards the end of the soft nematic solid regime and going into this phase transition with the plastic nematic solid, the order parameter increases along with the shape anisotropy. So you see this example here where there are these two things happening kind of simultaneously. And you see that's how you see these patterns with a high Q value, with a high order parameter value. We use the 2D vertex model to describe solid epithelial tissues, and additionally endow each cell with a nematic degree of freedom to account for the cellular active fibers. The mechanical energy is equation 1, which characterizes the shape, and then the rigidity, and this is just the mechanical energy needed to deform the cell. They use a Voronoi model, so they're using kind of calculating out the cell centroid based on these Voronoi cells. So it's like, you take a bunch of centroids and you find the mean distance between them, and then those are your cell boundaries. So and then the vertex positions evolve according to overdampened dynamics. Equation 2 is where we have the dynamics described by the dynamics of the vertices in contrast to the cell centroid-based approach used in the Voronoi model. The vertex positions R sub I revolve according to overdampened dynamics, according to equation 2, where you have a friction term that denotes the force induced by mechanical energy. Fi act is the active forces on the vertices induced by the cellular nematic stress. It's formed based on the definition of Cauchy's stress, which is here in 3, where phi act J, where phi act J represents the active nematic stress of cell J, and the summation is taken over the 3 edges connected to vertex I. So they're characterizing a trijunction here in terms of stresses. Here J and I denote the 2 neighbor cells. So J and I here, I think. Cell J in the summation is taken with 3 edges connected to vertex I. Here J and I denote the 2 neighbor cells sharing the interfacial edge E, and NCV is the unit's normal vector to edge E pointing from cell J to cell I. So they're basically constructing a network, and they're saying that it's directional in the sense that the stresses are moving from one cell to the other, and they're using the centroids as sort of the nodes of the network, and the edges are the stresses going to and from each cell. The active stress is created by cellular fibers in 4, where Q sub j is a symmetric and traceless tensor that describes the nematic order of cytoskeletal fibers, such as actin filaments and microtubules, organized within cell J. And then alpha is the activity. A positive negative sign of alpha corresponds to contractile, extensile cellular activity. The dynamics of Q sub j, which is symmetric, is at 5, where the two terms in this equation 5 describe the mechanical driving and intrinsic tendency of the nematic order, respectively. So I guess the mechanical driving is maybe this term with the beta in it, and then the mechanical driving of intrinsic tendency of nematic order is this last term here. The elastic stress on cell J is given by 6. The elastic stress on cell J is given by 6, where LE is the vector of edge E, and the summation runs over all edges of cell J. Its explicit form is given in equation A1. The coefficient beta denotes the strength of nematic alignment along the direction of deviatoric elastic stress. A negative or positive B drives QJ to a line parallel perpendicular to the stress. And so that means that, depending on this beta factor, it determines sort of the orientation of this nematic order, because you're getting a response to the order, sort of the orientation of the stress, and it's aligning itself appropriately. Given that cytoskeletal fibers often form supracellular structures, and that protrusions or filopodia can probe the local environment of a cell, so the cells aren't just these, polygons. They actually have like filopodia, and they have these weird shapes, and so the model doesn't really capture that. They're just kind of making an assumption here. They're building from this sort of a hexagonal or pentagonal model where you have like a 6-sided or 5-sided system with a centroid, and then they're just adding on like, okay, we can consider that there may be some weird deviations of that shape, and then factor that into the model.
Yeah, these are flat sheets, right? Because you have to have a proper adjoining sides, like a 5-sided hexagon, or it's not a hexagon. 5-sided has to go with a 7-sided figure, or you end up with 3D, like it'll buckle.
Yeah.
So these are all packed, as it were.
Yeah.
I guess that's what you'd call it. They're packed so that they're flat.
Right. Yeah, there are no gaps in between the cells, or no kind of misalignments. They're all kind of aligned. They'll kind of share boundaries, and then you get these trijunctions, as we saw. So that, but in real cell sheets, you wouldn't have that, right? You'd have like gaps. You might have weird alignments because they're always moving.
The ripples, like they'd be 3D.
Yeah.
Yeah.
And so then in the second term, the coefficient M controls the intrinsic tendency of cells to actively elongate and align with each other. A negative value corresponds to a non-elongated state with a round polygonal shape, while a positive value characterizes the state of an intrinsic cellular nematic order. Tau Q denotes the time scale of the nematic evolution, and it's proportional to a rotational friction. And then finally 7, which is the total stress on cell J, is stated in that equation there. So that's it. And then these are run in simulation, and then we get these results. So again, yeah, we get these results here. We have the plastic nematic solid. We have the onset of flow, a steady state flow. So you can see the difference here. they're just varying the alpha beta for the plastic nematic solid, and then varying the onset and steady state flow conditions. And then they show these defects. So they have the circles. The circles are plus half defects, and the triangles are negative half defects. So that then just shows you in their simulation where they're getting these defects. The lines, the red lines in here and the purple lines are flow field conditions. These are like the direction of the flows. So you can see they're changing direction a little bit. You have like areas where it's oriented right left or left right, and then top down, and then maybe some that are oriented diagonally. And they kind of maybe they determine these defects, maybe they don't. I don't necessarily see a correlation between where kind of you can see in some places where there's a change in direction uh, at the onset of flow and then the steady state flow where when the forces change direction, there are defects. So that's, I don't know much more about it than that. It's just kind of actually this is a velocity field, not a force field. So you have basically the shape axis and the velocity. The shape axis is in red, the velocity is in purple. Actually, in the velocity field, you see a little bit of unevenness along the edges, but that's just because they have to draw the lines around the edge of um, the matrix. So that's what we have there. That's, I think, one of their main results. What do you think, Susan?
I think you should go from this to the real world to
Yeah.
see what's going on, like compare. And I'm not sure what I should be studying in order to understand this thoroughly. It's couched in linear algebra as usual.
Yeah.
Yeah, I'm missing a piece of information. The linear algebra is sort of okay with me, but yeah, I just feel like I'm missing something.
Okay.
Yeah, I would like to know what. Like maybe I should send Machete an email saying, what do I need to study to thoroughly understand this?
Yeah.
I don't know, she might answer.
Yeah. Yeah, I don't know. Again, it's a model that starts from an idealized form, and then they use like Voronoi tilings, and they use centroids, and then they use these force and velocity fields so that you have these vectors. They influence then the shape of that matrix, and you can observe defects and things like that. But like in a real sheet, of course, you would have these like imperfections that would emerge from just like the cells moving against one another or them changing their shape. Because when they change their shape, they don't just move smoothly from like a hexagon to some sort of rounded object. They maybe have transitional states where they have there's slippage on the edges, and so you don't have these perfect tilings often. They're also moving in 3 dimensions, so they're moving over each other or under each other or something. So
yeah, there's hills and ripples
right.
as they go.
The displacement isn't uniform, I guess, is the lesson, especially higher sort of velocities. So yeah, I don't know.
Yeah, well, I would like to know more. It does fit in with what I was studying, so I don't know. I'd like to I might like to make a tensegrity model of that and then put it through its paces and see what happens.
Yeah.
If it would stand up and if the program wouldn't say that the building fell over, I might be able to do something.
Yeah.
I'd have to loosen the parameters up.
Yeah.
Of it
Building
somehow.
being the cell sheet.
Yeah, the building is the cell sheet, and the finite element programs think the cell sheet's a building. Anyway,
Right.
yeah.
Yeah. Well, Morgan and I were talking about an interesting paper that we saw. They were talking about it Saturday, where it was actually a model that someone had proposed for like a machine learning transformer, replacement for a machine learning transformer. And so it was this network. It was supposed to be like, it wasn't a neural network, but it was a network, certain properties that had like, it was a network with nodes, but it had elasticity in it. And the elasticity was supposed to represent learning. So, like, as the edges of the network kind of deformed, they changed their length or whatever. That was supposed to be like um, some sort of elasticity that corresponded to learning. But I was thinking, and I have to present on this more here, but I was thinking, well, this looks like a tensegrity model. And keep in mind that it wasn't at all, they hadn't at all thought about tensegrity, and it wasn't anything. It was a kind of a quasi physical model, but it did have a lot of those components. It was interesting.
Yeah, yeah. Well, if it ends up being a tensegrity, I can tell you it can be sometimes difficult to work with.
Yeah, yeah, yeah. It was like they kind of got at this tensegrity model from a different direction. It was weird, weird to see, but interesting.
I'd still like to know what odd electricity is, or elasticity is.
Odd elasticity.
Odd elasticity.
Oh, is that in the paper? I didn't.
I don't know if it was in this paper, but
Oh,
you can look it up. It just
okay.
seems strange to me, but
Seems odd.
yeah, it seems odd. Yeah, I don't know.
Yes.
Anyway, that's all I have this week. That's the thing I found.
Well, that's
And
great.
that
Yeah. Well, thank you for that. That was interesting. All right. So let me move on here. Yeah, so I wanted to talk about this idea of macromolecular crowding and thinking about this in cells. So last week we were talking about something like this, and some models of cells where there's this crowding inside the membrane, like so you have the nucleus, and then you have the cytoplasm, and then you have the cortex of the cell, and then it's in a lot of models that you'll see in textbooks, everything's very orderly. You have the organelles, and it's just like they show the space in interstitial space, and everything is very clear to see. But that's not how real cells are. What real cells do have is this macromolecular crowding. And so this is something I kind of looked up, and I used I don't remember what model this is, but Google's AI thing that comes up, and I want to look at the summary and then like go and find some papers. So this kind of describes macromolecular crowding. So macromolecular crowding in cells is modeled as a phenomenon where the high density of macromolecules, like proteins and DNA, occupy a significant portion of the cellular volume. And the estimate is like 5 to 40%. So remember, the cell is producing a lot of RNA molecules. It's producing mRNAs, miRNAs, and then it's also producing proteins, which are assembled at the ribosomes, or they're assembled in the mRNAs are assembled into amino acids at the ribosomes, and then proteins come from that. So it's a lot of stuff going on in the cytoplasm, and their estimate, or at least I guess from the literature, is something like 5 to 40%, depending on the cell, depending on the state of the cell. So I imagine that there are quiescent times when it's less crowded, and busy times when it's more crowded. But um, you know, so keep in mind that this is all happening sort of in this cytoplasm in between the nucleus and the cortex, and you get this, you know, um, basically it affects the dynamics of the cytoplasm. So we talked about condensates before in this meeting. So condensates are these sort of macromolecular clusters that sort of precipitate out of the cytoplasm. And it could contain proteins or mRNA or whatever. And those are kind of important in biology for various things. And so it's, this is an important thing to think about. So this part of the cellular volume, this sort of crowding, this high density of macromolecules impacts the function of other cellular components through steric and thermodynamic effects. So then they talk about models, how they use concepts like excluded volume to describe how crowding can do three things. The first, it's alter reaction rates by favoring reactions that result in more compact molecules. Two, affect diffusion, which can speed up some reactions while hindering others. And three, influence cell signaling by affecting the concentration of reactants available to membrane proteins. So cell signaling is another part of this, where you need to have signals coming into the cell from the receptors, and then they go down into the nucleus, those signals, and then they affect gene expression, or they can also affect the metabolic state of the cell. But of course, when you have this sort of crowding in the cytoplasm, it essentially is like kind of like a cloudiness in the sky. Like we're on the planet and we have an atmosphere, which is kind of like our cytoplasm for the planet. And well, it's a bad analogy, but you get my point. The more sort of more clouds there are in the sky, maybe the more haze there is in the sky, the harder it is to get sunlight, the harder it is to see the stars at night, that sort of thing. And so this can affect, in this case, it can affect a lot of things going on. Anything from reaction rates to diffusion to cell signaling.
Sorry.
Okay. So this was, let's see, this is a comment here.
Yeah, that's the other article. It's sort of a cellular crowding thing in a way, because you're getting phase separation between groups of molecules. It's sort of a molecule sorting, I guess, instead of cell sorting. It's that's how I think of it anyway.
Yeah. That's great. Yeah, thank you for that. So that's a poster.
Yeah, it's a poster, but there's a lot of other articles about the phase separation of interfaces inside cells. Like it's a whole topic.
Yeah. So yeah, then they have so that's that, and then people use computational and physics-based models to simulate this. So there's some key concepts: excluded volume, which is where the volume occupied by macromolecules prevents other molecules from occupying that same space. So they're basically like, they have a finite amount of space. You have molecules, once they're there, there's a rate limiting effect where other molecules can't be there. So if you have crowding, that just limits the number of molecules that can be in a certain place, in a certain location. And there's volume fraction, which represents the proportion of total cell volume occupied by macromolecules, typically 0.15 to 0.2 in a healthy cell. Models show that an optimal crowding level exists to maximize overall reaction rates without becoming diffusion limited. So, if you have diffusion, if you had if everything diffused, would reduce sort of the the effect of that crowding. So if every macromolecule diffused evenly across the cytoplasm, you would have, maybe you wouldn't have this crowding, but the way it tends to work is that you have these precipitates, you have these clusters that can interfere with processes, and you can model this as sort of like a diffusion limited system. And then diffusion effects themselves. So the presence of large macromolecules hinders the movement of other molecules. So again, if they're there, they can hinder the movement of molecules, especially if they're in a place where that molecule needs to be. The effect is dependent on the size and concentration of crowders. So small crowders can sometimes slow down diffusion more than large ones at the same volume fraction due to a larger surface area effect. So that's that one. And then this talks a little bit about how models are used for gene expression, for protein folding, for cell signaling, and in vitro models. And then, of course, there's some model-based simulations where you get these simulations of crowders. You get field effects observed. You get ion flux across cell membranes, those kinds of models. That show crowding makes the association of soluble macromolecules with membrane proteins much more sensitive to changes in cell volume than predicted by simple mass action principles. So, again, uh, we have to think about kind of how these things are distributed in the cytoplasm to understand their effects, their true effects. This is a picture of the JCVI Syn 3.0 synthetic cell. This is a model of that cell. The JCVI Syn 3.0 is a bacterial cell, so it's not a eukaryotic cell. And they model this crowding in this cell. This cell has a lot of crowding in the cytoplasm. So this is a digital model of the cell. It's kind of like the cell cortex is like a half shell, and then you have the molecular component kind of spewing out. So it's like cutting away the cortex, and you see the inside of the cell, and you can see all this crowding. So this is an accurate representation of this crowding. And this isn't a prokaryotic cell, mind you, so it's not the same as a eukaryotic cell. But you would see a lot of this sort of crowding. So you can't even see the nucleus. It's just kind of like all of these proteins and other macromolecules kind of in formation here. So it's just visualizing what this looks like. So I'm going to go through a couple of articles where they've talked about macromolecular crowding and what they're thinking about in terms of what it is and how we can, utilize it as a biological principle. So this paper is underestimated role of macromolecular crowding in bioengineered in vitro models of health and disease. And so macromolecular crowding, or MMC, is a ubiquitous phenomenon in biological systems that is largely overlooked in bioengineered in vitro cellular models. This comprehensive review examines the significant impact of both intracellular and extracellular MMC on cellular and molecular processes under physiological and pathological conditions. By synthesizing current knowledge and identifying critical gaps in our understanding of this review highlights the need to incorporate crowding into the development of in vitro models for studying health and diseases, as well as for drug discovery platforms. The pervasive nature of MMC in biological systems underscores its potential importance in various physiological and pathological processes, including protein aggregation disorders, cancer, and vascular diseases. So they're thinking about this macromolecular crowding when it's pathological, when it does bad things to the cell. And so you can imagine how that could happen, not just with the constraints I mentioned in the summary, but also kind of like if there's just way too much stuff in the cell. And so it interferes with like translation, or it interferes with um, metabolic processes. So it interferes with mitochondrial function, things like that. Recognizing ubiquitous influence of MMC could open new avenues for therapeutic interventions and deepen our understanding of fundamental biological processes. The second paper is on sort of molecular crowding at the origin of life. This is molecular crowding in early evolution. And this is a short abstract. So they're doing their experiments in protocells, which are vesicles. And so they don't have the same properties as like eukaryotic cells or even prokaryotic cells. There's basically these containers that you can put proteins into, and they behave, kind of like regular cells in the sense that they can perform metabolic functions and things like that. And so the environment of protocells might have been crowded with small molecules and functional and nonspecific polymers. So this is talking about the protocells at the beginning of life, where you didn't really have a cell yet. You had cells as basically things that were enclosed in a volume by some membrane. And, it wasn't like as complex as even the prokaryotic cells we see today. They were protocells. And so life was just trying to figure out what a cell should look like. And this is kind of where this crowding may have come from, where you have these molecules that are doing things in the cell as opposed to out in the environment. So they're protected by the membrane. Those protocells then can serve as a selective mechanism. So whatever's inside the cell can get things from outside the cell, but it's through, maybe like pores or through receptors or things like that. So there's signaling, there's a selectivity. And so that's basically kind of where they're going with this. We always had the crowding, but then we got sort of the more complex cells later. So the crowding's always been with us. And maybe it's even an artifact of these early stages in cell evolution. In addition to altering conformational equilibria, affecting reaction rates, and changing the structure and activity of water, crowding might have enhanced the capabilities of protocells for evolutionary innovation through the creation of extended neutral networks in the fitness landscape. So neutral networks are sort of a tool that people use to look at different equivalent states of, evolutionary change. So in terms of fitness, there are these networks that are sort of equivalent in terms of their fitness. Systems can explore these networks, different states in the network, and you can describe them using a neutral network. I'm not going to get too much more into that, but basically, the idea here is that you can have, you can explore the space more when you have these systems that can, help the protocell along and, give it adaptive capacities. Okay, so that's that one. The next paper here is this is the molecular crowding, the history and development of a scientific paradigm. This was in a special issue of Chemical Reviews on Molecular Crowding. So if you're interested, there is a special issue on this. And this is the abstract. So this is, I'll get into the text abstract and the visual abstract later. It is now generally accepted that macromolecules do not act in isolation, but live in a crowded environment. That is, an environment populated by numerous different molecules. The field of molecular crowding has its origins in the far 80s, I guess late 80s, but became accepted only by the end of the 90s. In the present issue, we discuss various aspects that are influenced by crowding and the need to consider its effects. This review is meant as an introduction to the theme and an analysis of evolution of the crowding concept through time, from colloidal and polymer physics, as we saw in the vesicle paper, to a more biological perspective. So this starts off as like a colloidal phenomena in vesicles. These, macromolecules are sort of in there as, kind of the functional show of the vesicle. As the functional show of the vesicle. And then later on, when you have cells with like nuclei and different types of functions. You then have these, they become things like condensates and other things, and they serve a wider range of biological functions. So this is their graphical abstract. Viscosity, so this is where they kind of talk about the role of diffusion here. You have viscosity, volume exclusion, and molecular interactions which affect diffusion. They have these condensates and other macromolecules in the cell. They're diffusing outward and they're getting more dense. And then these are the factors that are sort of influencing that. So whatever kind of medium constitutes the cytoplasm is going to affect diffusion. If there are a lot of things in the membrane, or if there are things clustered, say along the edge or near some organelle, then you can't also be there. So it's limiting as to what can be where. And then when you have more density in terms of what's inside there, it's harder for macromolecules to move. So they get sort of jammed inside that cytoplasm. And of course, then we're affected by protein folding, stability, and aggregation. So macromolecules aggregate into these condensates. It sort of opens up space for other macromolecules to migrate to diffuse. Stability is where do you have like a stable environment? Is there stability of the macromolecules? Is there stability in terms of the the function? And then aggregation, which we just mentioned. This paper is macromolecule crowding in vitro, in vivo, and in between. And so this just kind of talks about these heterogeneous and high volume spaces. So this is talking about biochemical processes. Biochemical processes take place in heterogeneous and highly volume occupied or crowded environments that can considerably influence the reactivity and distribution of participating macromolecules. Here we summarize the thermodynamic consequences of excluded volume and longer-range non-specific intermolecular interactions for macromolecular reactions in volume-occupied media. In addition, we summarize and compare the information content of studies of crowding in vitro and in vivo. We emphasize the importance of characterizing behavior not only of labeled tracer macromolecules, but also the composition and behavior of unlabeled macromolecules in the immediate vicinity of the tracer. So they're looking at things that we might be able to trace and see where they end up in the cell, and then things around it. Finally, we propose strategies for extending quantitative analysis of crowding in simple model systems to increasingly complex media up to and including intact cells. So, using quantitative analyses to sort of characterize and analyze this crowding, because it is important biologically. And again, when we usually build models, we leave out stuff like this. We just say, well, there's a density of macromolecules and it increases, and then maybe, there's an upper bound to that, and everything is fine. But it actually is, in and of itself, very much part of the system, and it affects how the system operates. Okay, I think that's it. I think I had those 4 papers and the summary. And, there are other images you'll see of macromolecular crowding. Like, people have kind of thought about this for a while. It's sort of like is a shorthand for the complexity of cells, right? Like, you have this stuff in the cytoplasm, it's packed with things. There are a lot of things going on in the cell outside of the nucleus. Of course, inside the nucleus, we also have condensates. We also have things like that. But yeah, so it's something that a lot of models don't really consider, and it affects the mechanics of the cell. So that's another thing we can put on that list of things that a cell should have. It should have a centroid. It should have boundaries, but it also should have stuff inside of it.
That's what my model didn't have in it, is it didn't have fluid. And I asked, well, could I put fluid in it, please? And the professor who's dealing with my project said no. He
Well,
just said it complicates things, but you can easily put water inside a form like that. So anyway,
I think
what
maybe
else can I say?
the concern was like,
Somebody, yeah, could somebody answer that? Anyway, what was I going to say? Oh, someone did. They took a truss and they put water in it, and it slowed down its movements. So that might be a good thing to. That might have helped its stability. So
right.
That's another thing I could try with my model, is to put water inside this truss.
Yeah.
It seemed to help its oscillations. Like, it oscillates very fast without the water, and slower.
Right.
Slower frequency with water in the model.
So yeah, I think there is. Well, what Susan was talking about, I think there was a concern that you don't want to model fluid dynamics for every cell in the simulation. It's likely to be an isotropic, within the cell, and then heterogeneous across cells. And I don't know. I don't think we're dealing with anything more than like basically laminar flows within a cell. Although, as you saw with the velocities, the velocity fields, as they're changing, you might get local turbulence. But that's something that is not. That's something we can only model so much, I guess. Okay. So, do we have anything else before we go?
Yeah, I should put it all in a supercomputer and see what happens. It'll crash, yes.
Oh, yeah,
Put a tensegrity in a supercomputer and it'll crash.
Yeah. Right.
Anyway, that's all. That's all.
All right.
I had my tensegrities hang up my computer. They hung it up. It kept iterating 5,000 times, and I finally turned it off and turned it back on, and then that worked. Anyway.
Yeah. All right. All right. That's good. Well, thanks for attending. Thanks for the paper, Susan.
Okay.
Yeah. All right.
It's one I'm very interested in.
Yeah. So. Yeah. Right. See you next week.
Okay. Bye.
Bye. All right. So I'd like to do another paper. This one is from the bioRxiv. This is just published this week on the server. And this is talking about the Asgard archaebacteria. So an Asgard archaeon with internal membrane compartments. So as we recall from the 3-domain model of evolution, we have bacteria, archaebacteria, and eukarya. And of course, we're in eukarya, plants are in eukarya, fungi, yeasts, and all of the rest. But we're actually closer to archaebacteria evolutionarily than bacteria. That is, if you go back to Luca, which is the last universal common ancestor, this lineage that comes from Luca, bacteria derive first. They split off first, and they form their different components of that clade, such as green non-sulfur bacteria, gram-positive bacteria, cyanobacteria, so forth. The other lineage that remained then was the common ancestor of archaebacteria and eukaryotes. So there's a split between archaebacteria and eukaryotes. What this means is that archaebacteria are more similar to eukaryotes, and if you look at cells, those cells then are going to more closely resemble eukaryotic cells than bacterial cells, although they'll have components that are sort of basal or ancestral to eukaryotes. So that's kind of a background for this paper. And again, let's think about macromolecular crowding in the context of this paper as well. So let's go over the abstract. The emergence of eukaryotes from a merger between an archaeon and a bacterial cell 2 billion years ago involved a profound change of cellular organization. So that's the other component of this relationship, is that eukaryotic cells are basically the combination of archaebacteria and bacterial cell types. So this was the sort of the way in which eukaryotes sort of became eukaryotes. And this is well before there was multicellularity. Well, the order in which different features of the eukaryotic cell arose remain a matter of controversy. Close archaeal relatives of eukaryotes have recently been identified that possess homologs of eukaryotic trafficking machinery. So again, this is the sort of machinery that you see near the membrane, where you have receptors, and you have different pores, and you have things that are coming into the cell, and those things are trafficked to the nucleus, or they're trafficked to other parts of the cell. So these archaebacterial cells have homologs of the eukaryotic trafficking machinery, a complex cell architecture, and other traits. So these traits are very similar to the eukaryotic cell. And this is the one that they're going to talk about here. The members of this phylum, the Asgard archaea, the proto-Eukaryotic, however, lack internal membrane-bound compartments, and therefore have shed little light on the origins of the hallmark eukaryotic endomembrane system. So here we report the cell biological analysis of a member of Archaea, Candiditis, Eubarchaeum umbrocryptum, in enriched mixed microbial communities, possessing a small genome encoding few homologs of eukaryotic membrane remodeling machinery. The cells of the species in late-stage cultures resemble previously described Asgard archaea, with extensive cellular protrusions. So they're very similar to the Asgard archaea that people generally use, but they have different properties that are more eukaryotic. Surprisingly, during early stages of culture growth, this type of cell has fewer protrusions, but possesses numerous intracellular vesicles, most of which have a luminal surface that morphologically resembles the outer coat of the plasma membrane. These data alter our view of eukaryogenesis by identifying a close archaeal relative of eukaryotes with a regulated endomembrane system. So we can look at light microscopy of this. So this is kind of looking at the properties of the cell. All right, so this is the cell body. This is the nucleoid, the cytoskeleton. You can see the protrusions out here. So you have these protrusions. So this is the cell body. This is a protrusion that comes around like this, and then it's meeting up with the cell. So the protrusions are generally used to move, like filopodia, or they could be used to sort of do other things in that intracellular space. G is showing a little bit more of the ribosomes, so they identify ribosomes in the cell. Their cytoskeleton is here, and the protrusions are here. So you see there are multiple protrusions. This is a segmentation model where they segment out the different components. They have the cytoskeleton, the ribosomes, the membrane, and the cell surface. This is more segmentation where they see more elements of the cytoskeleton. They see cytosol in here, ribosomes, and then the cell surface with protrusions. And again, the cytoskeleton, the ribosomes, the cell surface, the protrusions, granules here. So they're identifying a lot of components of the eukaryotic cell in these cells. This is a vesicle here. So in the light microscopy, you have a vesicle. You see the vesicles moving around inside the cell, trafficking things from the membrane into the inner parts of the cell. let's look at more light microscopy. This is an early stage cell culture. So this is just showing, the cells as they're young, and they have a lot of the features that are indicative of a healthy cell. So this is the protrusions once again. You have ribosomes identified. You have carbon support. You have vesicles here, cytoskeleton here. So this is the membrane, but the cytoskeleton covers the membrane. So they're trying to image basically the cytoskeleton to show kind of what that looks like. So again, this is early stage culture. Here's the nucleoid, there's a cytoskeleton, vesicles, and then you can see the segmented model where you have the vesicles, the cytosol, the cytoskeleton, the ribosomes, and then the outer membrane, which is the cell surface here, and the outer membrane here. Okay, so that's the segmentation model. And then this is, these are and so then these are ribosomes. So you see here the ribosomes along the edge. They look like little orange rings. And if you go here, you can see that their images here and then the model over here. So these are the ribosomes. You have the 50 subunit and the 30 subunit. So this is like in a eukaryote, in a prokaryote, you have like a 70S, you have 80S in eukaryotes. So the 50 subunit is here, 30 subunit is down here. The proteins are in orange, and the rRNA is in blue. So you see how the structure looks here. There's the identified exit tunnel. So, the mRNA goes into this tunnel where it's then transcribed into amino acids and then turned into proteins. And then there's an exit tunnel that comes out that gives the output of the amino acids. So this is, the basic structure. They're identifying all these components. There are also binding sites. There's the tRNA E site and the tRNA P site. They can also identify those. So they're just modeling this from these images. And so here they're looking at vesicle diameters. So here they're using cryo imaging. So they're using cryo-electron tomography to look at the different vesicles and their sizes. So you just have them arranged by size here, by diameter, and so you can then see kind of the distribution of vesicle diameters in different parts and the diameter for certain parts of the vesicle. So you have the single inner and outer, you have their diameter nanometers, and you have the distributions. They're more single vesicles than double or ones in outer or inner. And then these are the segmentation models of these single vesicles. You have an inner, so the inner vesicles are these circles within, and then the outer vesicles are these circles on the outside. So they kind of clarify this here. So the outer vesicle here is in blue in the segmentation model. The inner vesicle is in orange. So you have vesicles where you have sort of a ring, which is the outer vesicle, and then vesicles within it, which is the inner vesicle. And then you have these single vesicles in purple. And I'm trying to see if they have any single vesicles here. They don't really have good examples of that. Basically, the single vesicle will look like this, just with the sort of the ring, and then the compound, I guess you can call them compound vesicles, are less common, but they have this kind of structure inside. And so this shows a phylogeny of thermoproteoa in Asgard, which is what we're looking at here, we have this is the model that we're talking about in the paper, the Hamdallah Archaea. And then that is actually pretty close to the eukaryote, much closer than some of these other cells here in Thermioprotea. You see that there's a greater complexity of Philopodia, for example. You have bacterial cells that are put next to it in comparison. So the Lochiarchaea and the Hamdallah Archaea have Philopodia. They have these protrusions. They have the inner cell body. They have the compound vesicles, and those are all things you see in eukaryotic cells. You have the protrusions here, the Philopodia. You have the compound vesicles, and you have other organelles, and you also have the cytoskeletal structure, which you see here.